Okay, so I have the privilege of starting this next session, and I very much appreciate Dr. Sibeli's talk. You know, it's a reminder that Mother Nature works on our own clock, right? We'd like to rush things, and we'd like to control things as much as we can, but nature has a way, as they said in Jurassic Park. What I'm going to share with you today is some recently published and unpublished work from my laboratory where we use biomaterials to try to accelerate or guide this process. No disclosures on the work that I'm going to share with you today. So I don't believe I've heard too much about biomaterials in this meeting so far. We've heard a lot of great talks about using, isolating, expanding, duplicating stem and progenitor cells for therapeutic applications, and of course, cloning. But we haven't really talked too much about the role of biomaterials to guide the function of these cells in the application that we intend. Now, in my laboratory, we're very much focused on that process. We design biomaterials for a host of applications. They can be implantable, injectable, responsive to the cells, and provide cues to the cells so that they will function as we wish. And this is just a sampling of some of the biomaterials that we have developed and applied across a host of animal models in my laboratory. So one of the types of biomaterials we use quite extensively, we and the royal we, are hydrogels. Hydrogels provide really remarkable flexibility and opportunities to control and tune cell behavior. And of course, they can fill irregular defects that occur in the patient population that we're trying to address. The first, I'm going to share with you today 2 quick vignettes about recent work that we've done to guide mesenchymal stem cells toward wound healing and also osteochondral tissue formation. And the first was thinking about the influence of degradability, the rate of degradability of a hydrogel platform. Typically, most work that you'll see in the literature chooses a hydrogel for application. They mix it with some cell population or a protein. They inject it in the tissue site and watch what happens. But we really wanted to understand how degradability of the hydrogel matrix may influence the function of entrapped mesenchymal stromal cell and endothelial cell heterotypic cell spheroids. So the way we went about this work, and this is work completed by a recent PhD student in the laboratory, Vicky Tai, she selected a synthetic platform, a PEG maleimide hydrogel platform. This is a widely accepted, approved, very recognized as safe biomaterial. It's had tremendous success in immunoisolation and islet transplantation for diabetes. She complexed this hydrogel platform with an RGD motif so that cells could interact and adhere to the platform. And then we cross-linked these hydrogels with a mixture of non-degradable and MMP-degradable peptides. So by changing this ratio of non-degradable and MMP-degradable peptides, we could develop a non-degradable hydrogel, a slow-degrading hydrogel, and a rapidly degrading hydrogel. And you can see here, we loaded these hydrogels with these endothelial cell MSC spheroids, which we know secrete MMPs, so that we would get degradation of this platform. This is a gross morphology view of these hydrogels after 1 day. They do imbibe water and they swell. So I'm going to show you just quickly some of the results from entrapping spheroids in these hydrogels. The paper was published about a month ago in Materials Today Bio. You can see here that when loading MSC EC spheroids in a fully degradable hydrogel cross-linked with this GPQA peptide, we see rapid sprouting after just a few days into the hydrogel environment. In a partially degradable hydrogel, so we have a ratio of a degradable cross-linker and a non-degradable cross-linker, we still see some sprouting, but not quite as impressive as the fully degradable. And in the non-degradable hydrogel, we see no spreading or outreach into the surrounding environment. Confocal microscopy gives us an idea of what these lumens look like, what these sprouts look like as they extend to the environment. And when we quantify these lumens, not surprisingly, we see greater extension into the hydrogel as a function of the speed of degradation. What I'm not showing you here today is we also saw remarkable increases in trophic factor production as a function of degradability. So I encourage you to look at our paper where we looked at a proteomic analysis and we see significant increases not only in the complexity of the angiogenic secretome secreted by these cells, but also the concentration of these potent molecules like VEGF. So this is a powerful platform to guide the trophic factor production of spheroids. We translated the efficacy of this approach with an ex vivo full thickness skin model. So we got cadaveric skin from a company, Genoskin from France. We made a reproducible wound into this defect with a soldering iron, and we filled these defects with slow, rapid, and non-degrading hydrogels. So I'm really showing you here this thickness of the epidermal layer. An untreated epidermal layer is very thin. This is what the epidermal layer looks like when it's unwounded. And you can see here the relative differences in this sort of moderately degrading hydrogel and a non-degrading hydrogel. When we quantify this, we do see significant increases in epithelialization and granulation tissue with the more degradable hydrogel than the non-degradable hydrogel. And so collectively, this tells us that degradation is something that we need to be thinking about. Disease states that may have elevated production of MMPs may dictate the function and the response of this approach, but we have a tunable platform to guide this process. In the second vignette, I want to share with you something that my lab and many others have been working on over the last 5 years or so, which is thinking about hydrogels in a slightly different way. Instead of just a bulk injectable hydrogel, thinking about making small granular pieces of hydrogel and linking those together. So this approach of these so-called granular hydrogels has a number of advantages compared to typical bulk hydrogels. So the mesh size in bulk hydrogels is on the nanometer diameter, much smaller than mammalian cells. So cells are entrapped. They can't move and spread out until they remodel that local microenvironment, therefore slowing the inherent process of tissue healing. Whereas microgel-based scaffolds have a macroporous volume in between these microbeads, so cells can begin spreading and proliferating immediately. The stiffness and porosity characteristics of bulk hydrogels are coupled. You can't decouple these to understand the influence of these very important parameters, but in microgels, they're very much decoupled, so it gives you increased flexibility to explore this. And of course, bulk hydrogels, you're somewhat limited in the tunability of these aspects, but in microgels, you can combine these individual pieces like Legos in whatever manner that you want. So another PhD student who just finished in the laboratory, Dr. Jeremy Lowen, utilized a microfluidics-based approach to make these microgel-based platforms, again, from a synthetic polymer, PEG vinyl sulfone. In this early work, he cross-linked these microgel beads with a non-degradable cross-linker, PEG-DT, and he could decorate the backbone of the PEG surface with peptides of interest. In particular, you can see with this microfluidic device, we can generate large numbers of these microgels that are very reproducible in diameter size based on the characteristics of the microfluidic device that we've created. And so Jeremy wanted to use this process to make so-called chondrogenic-inducing microgel beads and osteogenic-inducing microgel beads and put these together to address the significant problem of osteochondral tissue repair. And so in order to do this, he used an incoherent mimetic peptide, HAVDI, and a BMP2 knuckle peptide, DWEVA, all complexed with an adhesion motif, RGD. Okay, so we're presenting 3 peptides to the cells on this surface. One that is chondrogenic-inducing, and this combination of RGD and DWEVA is known to be osteogenic-inducing. The only thing that we changed in this process is not only the peptides that were presented, but the stiffness of the individual microgel beads. So we know that cartilage really likes a softer environment than bone, which wants a stiffer environment to induce mesenchymal stromal cells to those phenotypes. So based on changing the concentration of the polymer, we could get beads that were the same diameter, but different stiffnesses. And of course, we can use a photo cross-linkable peptide and link all these individual microgels together into a contiguous structure. And so the first data that I show you here is that chondrogenic microgels outperform bulk hydrogels with the same characteristics. So if we take PEG vinyl sulfone coupled with the HAVDI peptide, we entrap mesenchymal stromal cells in these hydrogels. We can see that we have a very low concentration of collagen that's produced, a very low amount of glycosaminoglycan that's produced. But in comparison, the microgel beads exhibit much greater production of these key indicators of chondrogenesis. And of course, just simply looking at Alcium blue staining, we see very apparent increases in Alcium blue staining and GAG concentration in the microgel-based platform as compared to the bulk hydrogel, which is commonly used throughout the field. Similarly, when Jeremy did the same approach but for osteogenic microgels, he saw really quite similar results. He saw increases in early markers of osteogenic differentiation, alkaline phosphatase activity in the microgel platform compared to the bulk hydrogel platform. But surprisingly, we did not see accumulation of calcium in this construct. And this may be because the platform really doesn't support the nucleation of calcium at this stage at 3 weeks. However, looking at osteocalcin staining in this platform, it's very apparent that the mesenchymal stromal cells are undergoing osteogenic differentiation far better than those cells entrapped in the bulk hydrogel. So then lastly, Jeremy combined these two hydrogels, these two microgel platforms, to make a chondrogenic and osteogenic phase. And the nice thing about this is that unlike a lot of osteochondral systems, this is a contiguous system. We did not experience or observe any delamination, which can occur when you have hydrogels of very different mechanical properties or different compositions, because these are formed from the same polymer, and they're cross-linked together. And so that's a significant advantage in the field. This work is unpublished. I'm certainly happy to talk with you more about it, but Jeremy adopted a process of spatial transcriptomics, and he's the first person that we've found in the literature to adopt spatial transcriptomics applications and protocols toward a musculoskeletal application. Most of these platforms are ideally equipped for soft tissues, but they do a terrible job for bone. And so Jeremy worked quite diligently to look at the expression of genes as a function of position. And you can certainly see that the upregulation of genes characteristic of osteogenesis are seen on the bony side of this construct, and they're downregulated in the cartilaginous side. And then those genes associated with cartilaginous differentiation are upregulated on the cartilage side and downregulated on the bone side. And so in conclusion, what I've shown you today in these two brief vignettes is that matrix degradability significantly influences spheroid outgrowth into the surrounding microenvironment. This is a powerful tool to dictate and regulate the rate of tissue invasion and tissue repair in a variety of different disease models. And secondly, I showed you that microgel stiffness and peptide presentation are potent cues to guide mesenchymal stromal cell differentiation toward two lineages in the same construct. So now we're looking forward to translating this approach to animal models of osteochondral defects. And so with that, I'd like to thank the people in the group who did the work and, of course, the funding sources. I'm happy to take questions if I have time.
Thank you so much. We can take 2 questions. The next speaker can already go and set up. Lacey?
Kent, very nice presentation. Yesterday's presentation was also, I never made a comment, but it was exceptional as well. So this cell spreading that we see as a function of the degree of degradability, I guess, of the substrate, does degradability affect stiffness, or are the two completely independent of each other?
Yeah, so that's a great question. Certainly, as the material degrades, it does get softer in these PEG systems. You know, we did some mechanistic studies, which are included in the paper, where we blocked degradation with a GM6001 MMP inhibitor. We see that the spheroids actually get really frustrated. They remain metabolically active. Alamar blue activity is really high, but they get frustrated, right? And they can't sort of break through. We do see that the stiffness is maintained when we block MMP activation in that process. So as the cells are spreading, proliferating, producing more MMPs, the stiffness decreases. Our hope is that the cells are secreting ECM to balance this process. And so our next work will be in animal studies in collaboration with our colleague Aijin Wang to think about how we can treat full thickness defects with these approaches.
Any more questions? Yes.
Great presentation and very fascinating work. I was just wondering, how exactly was your graduate student able to conduct spatial transcriptomics on that tissue?
So we have a GeoMX platform at UC Davis, newly acquired. So this is about a $500,000 platform. The way that this process is done, you take histological sections of your construct, and then there are minute biopsies as a function of position that collect tissues and do RNA sequencing as a function of position. So you have to treat these constructs with specific probes so that they can be identified with a barcode matching system, and then you run RNA sequencing at that location. So it's not trivial. It's much harder than we thought it would be.
Thank you so much.
All right,
Ken.
thank you so much. I appreciate it.
Look at me from